fourth major tissue of the body, we have the nervous tissue. Okay, to review, we have the first one, epithelial tissue. Second one, we have the connective tissue. And the third one is the muscular tissue. And the fourth is the nervous tissue. The topics that we are going to tackle, we'll start first with the introduction. So we go into the function, the division, the composition of the nervous tissue. Then we go into the nerve cells, their morphology, characteristics, and classifications. Then inserted here will be the terminal endings because these are found in some sensory nerve cells. Then we go into the axons and the dendrites. These are the processes of nerve cells. Then lastly, we have the neuroglial cells with the different classification, characteristics, and function, which is in another set of PowerPoint. That will be our next set of PowerPoint, the neuroglial cells, or the neuroglia itself. Then another set of PowerPoint, we go into the central nervous system. We'll tackle here the brain and the spinal cord and the meninges that covers both the brain and spinal cord. Then lastly, that will be another set of PowerPoint. We'll go into the peripheral nervous system. Under this, we'll go into the ganglion or ganglia, the synapse, the nerve bundle, and the nerve fibers in the cranial, spinal, and autonomic nerves. We'll go into their histological characteristics, of course. Okay, so we go into the introductory portion of the nervous tissue, which I mentioned that this is the fourth major tissue of the body. And take note, the most highly differentiated tissue and the most complex. And this tissue is the primary tissue of conduction, coordination, sensation, and communication. The nervous tissue have four functions. We have communication, mental activity, control and coordination, and homeostasis. So in communication, the nervous tissue, or specifically the nerve cell, has the ability to respond to any stimulus in the environment, and this is conducted in the nerve cell as an impulse or electrical impulse and is carried from one cell to another cell to the brain for proper processing. Then we have a mental activity. It provides the structural and chemical basis of conscious experience. That's why we're able to be aware of our environment. What is happening around us, it's because of our nervous tissue. Then we have control and coordination of muscles and glands and other body parts. Now, in muscles, there are certain function of the muscles or certain contraction of the muscle which we cannot control, like the beating of the heart or the peristaltic movement of the digestive system. And also the glands in the body, they're also under the control of our nervous tissue. It's because of the chemical substances that must be released in certain amounts for the proper functioning of the body. And for homeostasis, the nervous tissue contribute so much in the internal environment of the body because of this control in some function of the organs and structures of the human body. The nervous tissue or the nervous system in the body is divided into two major parts. We have the central nervous tissue, which composes the brain and spinal cord, and the peripheral nervous tissue or peripheral nervous system. Included here is the autonomic nervous system. So here we will tackle about the ganglia, about the plexus, and all of the nerve fibers. Okay, so let's now go into the composition of the nervous tissue. It is composed of nerve cells or neurons. So that is the term for nerve cells. We have neurons. Take note, the main conducting elements. And these nerve cells is supported by neuroglia, the non-conducting elements or supporting tissue of the nervous tissue. Okay, so the neuroglia is composed of neuroglial cells, loose areolar connective tissue, and blood vessels, which we call nervi vasorum. The nerve cells or the neurons are described to be independent structural and functional units with complex morphologic characteristics. So, the nerve cells could function alone or independently or could function as a group or as a functional units. The nerve cells are composed of the cell body or soma or pericardion, and two processes. We have the dendrites and the axon. The nerve cell or the neuron receive information through the dendrites and cell bodies where it is integrated and then transmit this information onward via the axon. So in the illustration, you can see the red arrows wherein information is received by the dendrites in the upper processes and is forwarded towards the axon, then towards the brain for proper processing. This is another illustration of the complex structure or complex morphology of a nerve cell. But I would like you to focus on the terminal parts of the axon, which branches out to have a button-like structure as its ending. Now, in this illustration, it seems that the button-like structure of the axon is in direct contact with the receptor cell, which is here a skeletal muscle. But there is no direct contact with the receptor cell and the button-like structure or terminal ending of the axon. So which is illustrated here. So I would like you to focus on the lower portion wherein the terminal ending or terminal button-like structure of the axon is in contact with a receptor cell. Take note, there is no direct contact between the terminal button and the receptor cell. No, there is always a gap which we call synapse. So this is a summary of the composition of a nerve cell or composition of a neuron. So we have the soma or the cell bodies with its structure inside. Then we have the dendrites and the axon. So I would like you to take note that in the axon, it is mostly microtubules and neurofibrils. We'll go into that later on. And in the soma, we have the mitochondria and other structures. So here we have the endoplasmic reticulum. We go now into the cell body or the soma or the perikaryon of a nerve cell. So the cell body or the soma is made up of neuroplasm or a homologue of the cytoplasm of other cells in the body. The cell body or the soma represents the trophic center for the whole nerve cell. It has receptive capabilities. It receives a great number of nerve endings that convey excitatory or inhibitory stimuli generated in other nerve cells. Here we have an illustration of the soma that is receiving many terminal endings from other cells. The nucleus of a nerve cell is commonly spherical in shape, but sometimes they could be oblong in shape, and they are unusually large, euchromatic, pale staining with prominent nucleolus, making the nucleus its characteristic owl's eye or fish eye appearance. Meaning, as we see in the illustration, there is a pale staining area around the nucleolus, making the nucleolus distinct or prominent. And this gives the distinct morphological characteristics of nucleus of nerve cell, owl's eye, or fish eye appearance. The nucleus is most often located at the center of the cell body. So commonly, the nerve cells in the body, the nucleus are often or commonly located centrally. But there is an exception. Some nerve cells in the body, the nucleus are found at the edge or eccentrically located. So we have here the Clark's column of the spinal cord, and in some sympathetic ganglia in the body. The chromatin inside the nucleus is finely dispersed, reflecting the intense synthetic activity of the cell. The ribosomes of nerve cells may appear as free ribosomes or as numerous polysomes, so they are in groups, which usually form rosette or rosette formation. When appropriate stain are used, rough endoplasmic reticulum and free ribosomes appear under the light microscope as basophilic granular areas called nasal bodies. So the presence of ribosomes in the neuroplasm of nerve cells makes the nerve cell appear granular under the microscope. And take note, the granular structures, they are basophilic granular structures, and they are often referred to as nasal bodies. The number of nasal bodies varies according to neuronal type and functional state, meaning the type of nerve cells and their function. The ribosomes are particularly abundant in large nerve cells such as the motor neurons. And these ribosomes or these nasal bodies are involved during nerve cell injury, which causes a reduction in their number in the cytoplasm or neuroplasm of nerve cell, referred to as chromatolysis. The next structure is the rough endoplasmic reticulum, which is highly developed in nerve cells. They are organized into aggregates of parallel cisternae and are involved in protein synthesis. The nasal substances or the ribosomes, they are usually absent in the axon hillock or in the implantation cone wherein the axon arises. The smooth endoplasmic reticulum are not well developed in nerve cells, so that's why they are less obvious, but they are abundant in numbers and widely distributed throughout the cell. The functional role in nerve cells remains unclear, but they are known to sequester calcium ions and to contain proteins, and may provide a pathway for their distribution throughout the cell. The GOLGI bodies are multiple parallel arrays of smooth cisternae arranged around the periphery of the nucleus, but they are not found in the axon and they are confined only in the pericardium, but they have the same function as that of the other organelles in other body cells. The mitochondria of nerve cells is more slender than in other body cells. They are scattered throughout the pericardium, especially more abundant in the axon terminal or in the terminal button. They have the same function as in other cells in the body, the powerhouse of the cell. But another function of the mitochondria, especially in the axon terminal, is that they are involved in the relay of the impulse from the nerve cell to the receptor cell. There are two structures that forms the cytoskeletal support of the nerve cells. We have the neurofilaments and the neurotubules. The neurofilaments, they are intermediate filaments with a diameter of 10 nanometer, and they are found abundantly in the pericardium and in both processes, the dendrites and the axon, but mostly in the axon. Now, they need a special staining method for them to be seen or to be visible under the microscope. We have the silver impregnation method or technique, and after the staining method, the neurofilaments will appear as a black outline under the microscope. The neurotubules, they are larger than the neurofilaments. They are 24 nanometer in diameter, and they are just the same as those found in any other cells in the body. One of their function is to transfer a substance within the nerve cell to another part of the cell. Together with the neurofilaments, the neurotubules will provide the cytoskeletal support for the nerve cell. Let's go now to the nerve cells or to the neurons. So that's why they are referred to as the most complex cell in the body, because the neurons and their processes are extremely variable in size and shape. So they are variable in shape, variable in size, variable in number and length of cell processes, but they have a common distinct characteristic owl's eye or fish eye appearance of the nucleus, and they also have variable sizes and numbers of nasal bodies in the neuroplasm, and we have the neurofibrils or neurofilaments in the neuroplasm. We go now to the classification of nerve cells. So one criterion is based on the size, shape, and number of their processes. So we have, for the first type, we have the unipolar nerve cells or pseudo unipolar nerve cells. These cells have a single process close to the pericardium, but which divides into two branches forming a T-shape, one branch extending to the peripheral ending and the other towards the CNS. So during development, the unipolar nerve cells, at first, only one process will come out from the soma, but further development, this process will divide into two, will branch out into two, forming a T-shaped formation. So this becomes now a pseudo unipolar nerve cell. The second type is the bipolar neurons or nerve cells. So they have two processes, one a dendron and an axon. The third type is the multipolar neurons or nerve cells, which are the most common type of nerve cells in the body. They have more than two cell processes, one being the axon and the other dendrites. The next four slides will give us an illustration of the different nerve cells or neurons based on the number of their processes. So we have here in this illustration the bipolar, the multipolar, and the pseudo unipolar nerve cells or neurons. The next slide, we have types of neuron. We have here multipolar neurons with red and blue arrow. So the red arrow will represent inputs and the blue arrow the output of the information from dendrites to cell body to the axon, then to the brain. Then we have here the bipolar neuron, two processes coming out from the cell body with the red arrow and the blue arrow for the flow of information. And lastly, we have here the unipolar nerve cell with, at first, it has a single process coming out from the cell body, but later on branches into a T-shaped formation with the red and the blue arrow for the flow of information. The next classification of nerve cell is based on their function. So under this classification, we have the motor neurons. They control effector organs such as muscle, fibers, exocrine, and endocrine glands. So because they cause an action for muscle, they cause contraction of the muscle, and for the glands, they cause secretion of substances. They are also referred to as effector neurons. The second type of nerve cells are the sensory neurons. They are involved in the reception of sensory stimuli from the environment and from within the body. So they are also referred to as effector neurons. The third one is the interneurons. They establish interrelationship among other neurons, forming a complex functional chains or circuits. They are also referred to as internocial neurons. Some sensory neurons have specialized terminal endings. The terminal endings could be in the dendrites or could be in the axon. Some describe them to have the free endings of sensory neurons ramify with the non-nervous tissue cells by means of specialized termination. So there is a collaboration between the nerve cells and the non-nervous tissue cells. Wait. We have groups of terminal endings in the epithelium. We have the free nerve endings for pain. We have the Merkel's disc for touch, the taste buds for taste, olfactory epithelium for smell. So we have these are actually what we call neuroepithelium of the body. We have the taste buds. So on our tongue, we have the taste pores. So under it, we have the cells for tasting different food. Now we have the gustatory cell that is in collaboration with the oral epithelium. So we have here also the nerve fibers that innervates the cell. So there are four types of taste. We have the bitter, salty, sweet, and sour. And the last one, the fifth, is actually a new additional to the four basic tastes. We have the umami taste. It is the taste of the monosodium glutamate, or if we are fond of using vitsin, no, in our food or in our cooking. So it is described to be a meaty, broth-like, savory taste of the food, umami food. Then we have the cells involved in the sense of smell. So we have the olfactory receptor cells. So these are the nerve cells that is in collaboration with the surrounding epithelial cells, which are columnar in shape. So we can smell different odors. Then we have the Merkel's disc. So these discs are associated with epithelial cells on the surface involved in sense of touch in the epithelium. So this could be found in the epidermis. Then we have the free nerve endings. So these terminal endings are found throughout the body so that we can feel pain. So just like the description free nerve ending, so it is just like the ordinary ending of the nerve fibers spreading out through the different parts of the body, through the epithelium to the connective tissue for sense of pain. So we also have terminal endings in connective tissue. Again, we have the free nerve endings for pain, Meissner's corpuscles for touch, Ruffini's end organs for heat or warmth, Pacinier corpuscle for deep pressure, Krauss end bulb for cold. I would like to point out that the terminal ending in connective tissue for touch is Meissner's corpuscle, And in the epithelial tissue, it is Merkel's disc. So take note of that. We have here illustrations of the different terminal endings. So on the left side, upper and lower, it is Pacinian corpuscle for deep pressure found in the dermis or in the connective tissue. Then to the right, upper, we have the Krause and bulb for cold, and to the lower right, we have the Meissner's corpuscle for touch. You will notice that the terminal endings is enclosed in a capsule-like structure that is the collaboration of the nerve fibers and the non-nervous cells of the tissue, forming a capsule. The next three slides will show us an enlarged illustration of the different terminal endings. So we have here Pacinian corpuscle followed by the Meissner's corpuscle in the connective tissue or in the dermis of the skin, and followed by Ruffini's end organ for warmth and for hot temperature. We now go into the terminal endings in muscle. So we have the neuromuscular spindle, so between the nerve and the muscle. So it's called the neuromuscular spindle because of the shape, no spindle shape formation of the terminal endings. So these terminal endings for sensation of position and movement, for posture and basic adjustment of skeletal muscle. So we have here the spindle shape formation of the terminal ending, the neuromuscular spindle. So usually they could be found in inside the muscle, between muscle fibers. They are buried among the extrafusal fiber of the muscle, or they could sense if the muscle is undergoing stretching to the brain. Then we have the motor nerve ending or motor end plate. So this is located in the junction between the axon and the muscle fiber for muscular contraction. Then we also have terminal endings in the tendons, forming again into a spindle shape structure or terminal endings. We call them the neurotendinous organ of Golgi or neurotendinous spindles for tension. So they are mostly found on the edge of the tendon that is attached near its attachment to the muscle fibers. So we have here a summary of the different illustration of the terminal endings both in the epithelium and in the connective tissue. So we have on the left side upper, we have the Pacinian corpuscle for warmth. On the right side, we have the Meissner's corpuscle for touch in the dermis. Then in both epidermis and connective tissue, in the middle illustrations, we have the free nerve endings. In the dermis, we have the Ruffini's end organ for warmth or for hot temperature. Then Krause and bulb for cold. And we have on the lower portion, we have now the different spindle shape or terminal endings found in the muscle or found in tendons. Let's now go into the different processes of nerve cells. So we take up first dendrites. So dendrites are multiple elongated processes specialized in receiving stimuli from the environment, from sensory epithelial cells, or from other nerve cells. So you can see that the dendrites are multiple branching surrounding the soma of the nerve cell. The axon is a single process specialized in generating or conducting nerve impulses from the soma to other cells, either to the next nerve cells or to the muscle cells or to glands. Now, I would like to point out that the distal portion of the axon is usually branched to form what we call the terminal arborization or telodendrion. And each branch of the axon of this network formation terminates on the next cell in dilatation called end bulbs or botons, which forms part of the synapse. So synapses transmit information to the next cell. Take note that both dendrites and axon are referred to collectively as nerve cell processes. There are 7 characteristics of a typical dendron. So number 1, it arises from a broad base from the soma of nerve cells. Presence of nisal bodies. Now the presence of nisal bodies in the dendrites or in a typical dendron gives them a granular appearance under the microscope. The dendrites or a typical dendron, they are short and stout processes at the beginning, rapidly becoming thinner due to the number of branching. Now, if ever they branch, they show profuse branching, similar to a branching of a tree. Very important characteristic is number 6, the presence of gemmules or thorny spines along the branches of a typical dendron or dendrite. So take note of this, gemmules or thorny spines. And if ever they branch proximal to the cell body or they branch near the soma or the cell body. For the typical axon, there are 7 characteristics. Number 1, it arises from a clear conical area devoid of nasal bodies called the axon hillock or implantation cone. Since this is a clear area, there is absence of nasal bodies. Now, what will be present will be the neurofibrils or neurofilaments, which gives the axon a fibrillar appearance under the microscope. Now, the axon is a long and slender process, gradually diminishing, and if ever they branch out, there are only few branches that is given off by the axon, and we refer to them as collateral. So collaterals are given off at right angles at the level of the nodes of Ranvier. Take note, there are no characteristic gemmules along the branches of the axon, and it ends up in a terminal arborization or telodendrion. If ever they branch out far or distal from the cell body. We have here an illustration of a nerve cell with the axon. So this will help us better to understand the morphology of the axon. So the axon will arise from an area in the soma or in the cell body of nerve cell that is devoid of micelle substances. We call the axon hillock or the implantation cone. Then we have here in the illustration branching of the axon we call collateral, and we have here at the terminal portion of the axon we have a network or we have an arborization of the nerve fibers or the axon fibers we call as the telodendrion, and in each branch we have the terminal button. Now also illustrated here is the sheath that protects the axon. We have the myelin sheath. We'll take this up when we reach the peripheral nerve system. And also, we have the alternating nodes here in the axon together with the myelin sheath. We have the node of Ranvier. So in summary, the nerve cell processes involve the dendrites and the axon, and both are referred to as nerve cell processes, and their primary function is reception of various stimuli, conduction of the stimuli, and synaptic transmission of the signal to subsequent neurons in the neural pathways or to muscle and glands. So number one is the function of the dendrites, two, both, and three, by the axon. Our next topic will be on the supporting tissue of the nervous system or the nervous tissue. We have the neuroglia, especially the neuroglial cells.